Hey everyone, today's lesson is going to be over viral reproduction, and our learning objective is we will describe viral reproduction, including the two viral reproduction cycles. So, we talked about viruses a while ago, so we know that viruses can't reproduce on their own, but that most viruses are cell specific. So, there are certain proteins that specifically bind to other cells for entrance into the cell to reproduce. Every single cell is different, so because of that, that means that there are only certain cells that viruses can actually attach to. So when we look at COVID, COVID is a virus that impacts only the respiratory cells in humans, and that is due to the specific surface proteins that are present. Now, this picture here is not exactly COVID, but it illustrates really well how you've got your virus right here, and it's got certain proteins that are surrounding the capsid, and this is your receptor on the cell right here. So you can see, obviously, they are pretty close to each other, and when they bind together, that enables this virus to be able to place its DNA inside. Now, these surface proteins are important because that is what enables a virus to impact a cell. If this cell did not have this specific receptor right here, then this virus could not impact that cell. So, with viral reproduction, we also know that viruses can be made of either DNA or RNA, but there's a group of viruses that we call retroviruses, which are made up of just RNA. Now, there are four main stages to viral reproduction, and those are entry, replication, release, and latency. Now, these are just a basic pattern, but it isn't a completely standard formula. We're going to go through the two cycles that viruses can undergo in order to be able to reproduce. And while they will use a combination of these four stages, it is not just going to be exactly this. These are just four basic stages that viruses can go through. So the first cycle that we're going to look at is called the lytic cycle. And so I've got a really good visual here for you to be able to see kind of how the lytic cycle is going. But as we go through this, I'm going to highlight the 5 stages of the lytic cycle. So the very first stage is simply attachment. So this is up on number 1, where you see that specific virus attached to the specific cell. Like we said earlier, there are proteins that enable the virus to be able to bind to a cell. So that is what's happening at this point, is that attachment. Then number 2 is entry. So at this point, that's when the genetic material is being pushed into the cell. So it is not the entire virus itself that is necessarily being taken it up by the cell. In a case of like a bacteriophage like this, it is only the genetic material that's actually being placed into the cell, and the rest of the virus actually stays outside. Number 3 is when they get to replication. So this is when the cell makes more of the viral DNA and proteins. So we know viruses can't reproduce on their own. They have to hijack another cell in order to be able to do that. So this is the part where they are using the cell's mechanisms in order to be able to create their own DNA and proteins for that phage or the virus genome that was placed within the cell. Then after that, the new viral particles assemble in a process we call assembly. So they have to create all of these parts again, and then they've got to come back together into a new phage or virus. And then finally, the last thing is that they get released. So the new viruses leave the host cell. And in the lytic cycle, they burst or kill the host cell. Okay, this is going to be very important to remember, is that they burst or kill. So you can see right here, they completely break apart this cell. So from attachment, entry, replication, assembly, to ultimately killing off this cell. The second cycle that we have to look at is the lysogenic cycle. So like I said, with the stages of viral replication earlier, there's going to be some things that are similar and different. But the 4 stages of the lysogenic cycle is, the first one is attachment again. So it's the exact same thing as what we saw last time. At some point, that virus has to attach to the cell. And then the second step is the exact same as well. There has to be entry. You can't start increasing or replicating your DNA if you don't actually enter the cell. Now, this part is where it's actually going to get a little bit different from the lytic cycle. So instead of actually synthesizing or copying its genetic material, in the lysogenic cycle, it actually integrates. So during this step, the viral DNA integrates into the host genome. So you can see right here, This little blue portion is actually the portion of DNA that came from the virus. It forces itself into this organism's DNA, so now it is actually a part of it. It's not distinct anymore, but is um, simply attached within this DNA of this organism. And then the last step in the lysogenic cycle is actually cellular division. So the host cell reproduces and copies the infected genome into the daughter cells. So if this is the cell right here, well, once it goes through mitosis, it's going to start creating two copies of that DNA. So now you have that viral DNA that's been replicated by the cell itself. So this time, there is no bursting or killing of the cell. In the lysogenic cycle, these viruses, their whole goal is to be able to reproduce within those specific cells in order to be able to increase their number that way. And so the the lysogenic cycle sometimes will continue on for a while until at certain points it can sometimes go back to the lytic cycle where it will start to actually affect the organism a little bit more, but sometimes it will just stay within the cell itself and do some sort of other process in that way. So that's it for what I have for today, and keep learning further.